would like to thank the organizers for this opportunity to contribute to this educational session, and thank you for everyone streaming this presentation. I have no disclosures relevant for this presentation. Precision cancer medicines are increasingly transforming patient care. This conventionally involves basic and translational scientists working together to develop new therapies and then advancing them for clinical development. This is sometimes depicted as a linear process from the lab to the clinical or bench to bedside. However, it is increasingly clear that it is more accurately viewed as an iterative cycle, with data flowing from the laboratory to the clinical setting and back to the lab as we learn from clinical experience. Functional diagnostic approaches, where potential treatments are tested on a sample from an individual patient, are being developed to guide individual patient care, and are an exciting and rapidly evolving part of this precision medicine ecosystem. In this educational session, I will focus my presentation on how preclinical studies from my research lab, as well as the work of many others, can help to inform the development of functional diagnostics. As part of our work, and in collaboration with teams at the Broad Institute and with other partners, we have launched the Cancer Dependency Map. This aims to use large-scale functional genomics to identify vulnerabilities, so-called dependencies, in every cancer cell. We believe that a map of cancer dependencies and the biomarkers that predict them could serve to help accelerate the development of precision cancer medicines. To assemble a dependency map, we are using large panels of cancer cell models, together with functional genomics approaches, such as large-scale drug sensitivity testing, to study cancer gene function, discover therapeutic biomarkers, and to identify new drug targets. To date, the Sanger and Broad Institutes together have generated data for over 1,700 cancer cell lines, including genomic, epigenetic, proteomic, and functional data sets. Coverage of the cell lines for each data type is shown in the image. It is important to note that we use this large collection of cancer models to have representation of different cancer molecular subtypes that occur in patient populations and underpin differential therapy response. We continue to expand these datasets through the development of new cancer models and their enhanced annotation with new molecular and functional data. An important component of this has been high-throughput drug sensitivity screens across our heterogeneous collection of cancer cell lines. As an example, the data shown here are IC50 values for 903 cell lines screened for their differential sensitivity to the BRAF inhibitor Dabrafenib. Each circle represents the sensitivity of an individual cell line, and this example highlights the range of sensitivities observed across the cell line collection to this particular drug. We integrate drug sensitivity data with genomic data to understand the molecular changes that underpin the range of drug sensitivities. For example, as shown on the left-hand side of the image, mutation of BRAF in cancer cell lines confers increased sensitivity to BRAF inhibitors, particularly in melanoma cell lines, where dabrafenib is clinically approved for the treatment of BRAF mutant tumors. In contrast, NRAS mutant melanoma cell lines are resistant to dabrafenib, consistent with what is observed clinically for these patients. Perhaps most interesting, BRAF mutations also occur in colorectal cancer, and, unlike melanoma, colorectal cancer cell lines are relatively insensitive to dabrafenib, mirroring the lack of clinical activity of BRAF inhibitors in BRAF mutant colorectal cancer patients. These examples demonstrate the power of large-scale drug screens to identify clinically relevant molecular markers of therapy response. They also showcase the impact of molecular background and, in some cases, tissue lineage on shaping response to therapy. Beyond this specific example, we have screened full dose response curves for over 1,400 compounds in 1,000 human cancer cell lines, generating in excess of 1 million dose response curves to link drug sensitivity with genomic alterations. In the study shown here, we correlated the sensitivity of hundreds of anti-cancer drugs with cancer driver mutations, identifying hundreds of putative biomarkers of sensitivity. The volcano plot on the left-hand side shows different biomarkers of drug sensitivity and resistance. These include the known association between EGFR mutations and response to EGFR inhibitors in lung cancer cells. In addition, this approach identified many new biomarkers of drug activity, many of which are poorly understood with our current knowledge and could contribute to the development of these drugs and targets. When we map biomarkers of drug sensitivity identified in cell lines onto sequencing data from patient tumors, we find that across different tissue types, a median of 50% of patient tumors have a genetic biomarker for drug sensitivity. This is highest for colorectal cancer and lowest for ovarian cancer. And this is consistent with data from clinical sequencing of patient tumors, indicating that up to 50% of patients have at least one candidate actionable mutation that might confer increased sensitivity to an existing drug. Now, our results are derived from cancer cell lines and so should be interpreted cautiously, and the effects observed in cell lines may not translate to meaningful clinical activity, and they also likely reflect the upper limit of current molecular interventions. Nonetheless, in the context of these caveats, our results suggest that there are likely to be more patients that could benefit from existing targeted therapies than currently do today. In the context of functional diagnostics, these data could also help to nominate drugs for testing and to interpret results generated from diagnostic assays. There are many unexplored potential cancer drug targets. For most proteins, there is not a drug that can be used for screening to validate it as a target. We are performing genome-wide CRISPR knockout screens in our cancer cell lines to identify and prioritize new synthetic lethal targets. A schematic of the experimental design is shown here. We performed screens in 325 cell lines and identified the subsets of genes which are required for cell fitness in specific contexts. We combined the fitness effect with the orthogonal information, including whether the gene is altered in patient tumors or aberrantly expressed, whether there is pathway level support for a target, and finally, whether there is a genomic biomarker linked with differential fitness effects observed in a subset of cell lines. We performed this analysis considering each cancer type individually, as well as combining cell lines from all cancer types. Together, this gave us ranked lists of targets based on a priority score for each cancer type and pan-cancer. We also overlaid the priority score with information about the tractability of a target for drug development. We identified over 600 candidate priority targets with different levels of evidence for tractability for drug development. Each point here represents a candidate priority target, their priority score, and tractability. Group 2 targets are a particularly interesting group because they represent new candidate targets with evidence supporting their tractability. Among these, we identified Werner helicase as a promising new target. Werner is indicated with an asterisk. Werner is selectively required for the fitness of microsatellite unstable colorectal and ovarian cancer cells, and when considering all cancer types together, as shown in the left-hand side panel. Werner knockout leads to profound chromosomal instability and apoptosis, and these effects are not observed in microsatellite stable cells. We also used mouse xenografts to show that Werner knockout leads to suppression of tumor growth in vivo. Based on these results, there is now interest from the pharmaceutical industry to develop Werner drug discovery programs. This study also demonstrated how CRISPR synthetic lethal screens can help to systematically and in an unbiased way nominate and prioritize diverse candidate oncology targets. It is widely recognized that cell lines have a number of shortcomings, including having adapted to culture in plastic and insufficient representation of some tissue and molecular subtypes. Historically, the development of new cancer cell lines has been very difficult, often taking many months with low success rates. However, recent advances in cell culture methods, and particularly the use of organoid cultures, have improved the efficiency of deriving cell culture models. Thus, it is now possible to derive tumor organoid cultures representing different tissue and molecular subtypes, as well as from different disease stages, with much greater efficiency than was previously possible. There are now a number of publications, such as those listed here, reporting the generation and characterization of organoid biobanks, including their use for modeling drug sensitivity. In aggregate, these studies demonstrate that organoids are genetically similar to the original tumor from which they were derived, that they can be used to model clinically relevant therapy responses, including recapitulating known biomarkers of response to targeted therapies. In retrospective studies, tumor organoids have been used to predict patient clinical responses to therapy. And co-culture systems incorporating components of the tissue stroma and immune cell populations have been described recently and used to model response to checkpoint inhibitors. The ability to generate models of patient-derived tumors much more efficiently is having a transformative effect on our ability to study disease mechanisms and therapy response, and more specifically, to generate models for functional diagnostic testing. We are creating a new collection of patient-derived tumor organoid cell models to supplement our 2D cell line collection. These are generated as part of an international effort called the Human Cancer Model Initiative, and all models are being made available as a community resource. In the UK, we have created a network of 9 clinical sites to access tissue for derivation. Tissue from surgical resections are couriered to the Sanger Institute, where organoids are derived in a dedicated facility. Following rigorous quality control, organoids are banked and sent to the repository ATCC for distribution, and enter into our downstream analysis pipelines for genomic analyses, drug testing, and CRISPR screening. To date, we have processed over 488 patient tissue samples from surgical specimens, and this has led to the successful derivation and banking of 136 organoid cultures from colon, pancreas, and esophageal cancers. The mean duration from derivation to banking is 104 days, and failure to propagate and low tumor cell numbers following derivation are the most frequent causes of banking failure. Our success rate is lower than what is sometimes reported in the literature, and we believe this is due to the fact that we take all samples received without any pre-screening and generate a bank of 25 cryovials for a derivation to be deemed a success. Thus, while organoid cultures represent a huge leap forward in our ability to develop cancer models, the time required to develop cultures may be prohibitive for some functional diagnostic approaches, and improved methods are needed to increase success rates, particularly when working with diverse samples. We have developed methods for drug screening organoids in 384-well plates, and I would like to share with you an early study that exemplifies our approach and some of the challenges involved. We derived a proof-of-concept biobank of 9 esophageal adenocarcinoma organoids together with linked clinical data. This was exciting because there are a lack of robust preclinical models for this poor prognosis patient population. Our clinical data included details of neoadjuvant chemotherapy regimes and the tumor response grade, or TRG, which is a score of how well the patient's tumor responded to therapy. Most patients did not respond to chemotherapy and had a TRG score of 4 or 5. And although there is typically insufficient tissue available for organoid derivation from patients with a good response, we did derive an organoid from one patient that responded well to chemotherapy and had a TRG score of 2. We profiled the sensitivity of the organoids to 24 clinically relevant targeted small molecule inhibitors. This heat map shows that we observed a range of sensitivities to the compounds, with some organoids sensitive to a significant number of drugs, indicated by the yellow squares. Compounds with a similar target had similar activity profiles across the different organoids. For example, ERBB2 inhibitors clustered together. The relatively small number of organoids screened prevents us performing a robust, systematic statistical analysis for genomic markers of sensitivity. However, we are able to make some observations based on prior knowledge. For instance, the only TP53 wild-type cell line in our panel is sensitive to Nutlin3A, and the 2 PI3 kinase mutant organoid cultures were preferentially sensitive to PI3 kinase inhibitors. We also tested the sensitivity of our organoid panel to the neoadjuvant chemotherapy regime the patient received and compared this to their TRG score. We used a combination of cisplatin and 5-FU, which were used at fixed concentrations together with increasing concentrations of epirubicin. As shown in the dose-response curves on the left-hand side, the majority of organoids were relatively resistant to the chemotherapies, even at high concentrations. But we did observe increased sensitivity for 2 organoids, despite having a high TRG grade, indicating that they did not respond clinically. The viability effect of different combinations are summarized in the bar plot. In the 2 sensitive organoids, there was benefit from a combination of drugs compared to epirubicin alone. Designing and interpreting these experiments is challenging. For example, when using a triple combination, it is difficult for us to know what concentrations and degree of response is clinically relevant. The TRG grade is also a subjective measure of response determined from radiography of the tumor, and there is a 2 to 3 month period between neoadjuvant therapy and surgical resection, and so clonal selection of the tumor may occur during this period. The one organoid in our collection from a patient with low TRG score, meaning they responded to chemotherapy, did not grow sufficiently well to perform drug testing, and so we are unable to compare the sensitivity of low and high TRG tumor organoids. The absence of a physiological tumor microenvironment may also affect these results. In a similar study, patient-matched organoids were evaluated to discriminate between standard of care chemotherapeutics for the treatment of metastatic colorectal cancer patients. Organoids were established before the start of treatment. Notably, organoids from patients with stable disease on irinotecan were more sensitive to irinotecan than organoids derived from patients with progressive disease. This is shown in the dose-response curve and scatter plot on the left. Patient-matched organoids were also predictive of clinical responses to a combination of 5-FU with irinotecan. In contrast, as shown on the right-hand side panel, patient-derived matched organoid cultures were not predictive of clinical responses to oxaliplatin for reasons that are not understood. In aggregate, there are multiple lines of evidence that organoids can capture clinically meaningful drug responses, both in the preclinical setting and as a functional diagnostic. But nonetheless, there are cases where patient-derived organoids do not recapitulate clinical responses. In summary, integrating preclinical discovery, functional diagnostics, and clinical experience are helping to deliver precision medicine for many more patients. Functional diagnostic approaches, particularly when linked with genomic analyses and patient follow-up data, could make an important contribution, not only for an individual patient's care, but by increasing our understanding of the molecular changes in cells that underpin clinical therapy response. This could contribute to the development of better therapeutic biomarkers. Our ability to grow patient-derived tumors and systematically perturb them is also empowering functional precision medicine. Nonetheless, better methods to efficiently acquire patient material suitable for testing and reducing variable culturing efficiency are needed. Furthermore, prospective studies are needed to better interpret and improve the predictive power of functional diagnostic approaches, so we understand which functional diagnostic test is predictive for each patient. I would like to close by thanking all of the people at the Sanger Institute involved in this work, as well as a fantastic group of collaborating laboratories, as well as my funders. Thank you for your attention.